cordial greetings throughout the Autonomous University of Baja California, the Faculty of Chemistry, Sciences, and Engineering. Today, we are pleased to share with you the Structural Biology Symposium, in which we will be joined by two excellent researchers, Professor Alastair Stewart and Dr. James Davies. We remind you that these seminars are intended to broaden the horizons for all your students, so that they can learn about greater range of career options. This time, the seminars focus on basic research in structural biology, where we hope you will be able to learn about the work of these two excellent researchers in the field of structural biology from the University of New South Wales. This is a top 20 university worldwide. A brief introduction of Professor Alastair Stewart. He completed his undergraduate degree at the University of Cambridge, England, graduating in 2007. He completed his PhD at the University of New South Wales, Australia, graduating in 2012. He currently has his own laboratory at the Dr. Chang Cardiology Research Institute in Sydney, Australia. He has published over 35 research articles, which probably is going to go to 40 at the end of this year. And he has over 1,700 citations. In the area of ATP synthase regulation, he is one of the top researchers, if not one of the top two. His work in the structure of bacterial ATP synthases has changed the way we understand the regulation of these enzymes, and he has helped advance our field greatly. It is my great honor to have him give a seminar in our university. I hope you can all enjoy his talk as much as I have enjoyed them in the past of several of these seminars. I now yield the microphone to Professor Alastair Stewart, if you would like to share the screen.
Thank you very much, Francisco. And apologies to start with, I had to reschedule. I ended up getting influenza, I think as we said. So is my screen sharing now? Yep. And Francisco recently visited us here in Sydney. We had a wonderful 3 months exploring the local area, exploring science, and yeah, having a wonderful time. It was wonderful to be here, and thank you very much for hosting us. So today, hopefully, I will be able to give you that working, and then put a laser pointer on. Hopefully, I'll be able to give you maybe about a half an hour talk about how we're able to use electron microscopy to understand certain things. So my lab and the way I was trained was from this idea of working on structure and structural biology. And at the end of the day, you know, I've only been in research for about 17 years now. Even during that time, we've used different structural techniques to be able to do things and see things in different ways. So when I started, I did something called X-ray crystallography, that many of you may have heard of, and this is where we're able to make crystals out of proteins or small molecules, and we expose them to X-rays, and that allows us to understand the structure of those molecules. And then after that, I did a small amount of NMR to try and understand, which Francisco's done much more than me, to understand how certain molecules work. And then around about 10 years ago, I was lucky enough to be involved with the two groups where we started working on electron microscopy. And electron microscopy was nothing new, but a series of things happened around about 10, 15 years ago, which really changed how we could use electron microscopy to look at things. So hopefully what I can show you today is how we can use electron microscope to look at molecules. And in our case, even setting up how we image those molecules to try and understand the steps of catalysis. So seeing catalysis in an electron microscope. So after I trained in electron microscopy, pretty much over in Singapore with a good friend of mine, Sarah Sandin, I was lucky enough to be able to buy our own electron microscope here back in about 2018. So this is us installing this electron microscope. It's not huge, but it's sort of, you know, weighs about the same as a car. I guess it's about the same size as a car. And it comes in one of these big sort of containers here where it has a series of computers. It has machinery here to kind of fire electrons down through a column, and then you put your sample in here and you can image at the bottom. And this is the shell of it, but if you look how it really is installed in our university here, you can see it comes with this sort of fancy box around the outside. And this is me and Cruz, quite cheesily looking for a photograph here. But what you can see again here is you have this electron microscope here. This is a column where electrons run through here, get fired down by a gun. They get magnified on a sample and go on the bottom. And a couple of things that have happened over the last 20 years have really changed how we can use this. The first one here is actually on the left here. I don't know how big your screen is, but there's something called an autoloader. And this has allowed us to really increase the throughput of how we're able to look at these samples, because we can load sort of 12 samples at a time and use a robot to put those inside this electron microscope imaging. And the other one, that rather than using staining methods, you can see here I'm pouring out some liquid nitrogen. We look at these samples in cryogenic conditions. So we often refer to this as cryo-electron microscopy. So rather than looking at stained molecules, which would really reduce our resolution, we're able to look at frozen molecules, which is able to keep lots of information and allow us to identify where particular atoms or groups are in our molecules. So this technique of cryo-electron microscopy and single particle analysis was awarded a Nobel Prize about eight years ago now, 2017. And they gave this to Jacques Dubochet, Joachim Frank, and Richard Henderson for how they drove this methodology of electron microscopy from quite a low resolution method to just whether you might be looking at stained sections or muscle, for example, was very much used 50, 60 years ago in order to use electron microscopy. But there they were just really looking at cellular architecture. And what these three people did, and many others actually, was able to develop new ways of using this electron microscope to see this high resolution structure determination of biological molecules in solution. So they could see these molecules at high resolution, and they could also see them in what they're referring to as the solution phase here. Because at least until this point, generally when we've done crystallography, we were looking at things in a solid phase or a crystal. So we put all these molecules together to make a crystal, and that sometimes meant that we couldn't access, as you'll see in this talk, a particular state or particular understanding of that. So we look on the right here. I've got a video, but I'm just going to turn this laser pointer off so I can stop and start it, because I don't know how well it will present over the internet. But the Laboratory of Molecular Biology, actually, where Richard Henderson was, made a nice video that describes how this method of cryo-electron microscopy works, single particle analysis. So you'd start with a tube, in this case like an Eppendorf, and inside that Eppendorf you'd have a solution, and inside that solution they've shown here a series of protein molecules. So this is usually what we do, is we purify protein molecules. In a cell you might have 20-something thousand different proteins, and we try and pull those protein molecules out to make a solution which is generally only one of these proteins, or a protein complex sometimes. So inside this Eppendorf, you had a series of different protein molecules, and because they're in solution, they're all moving around in different orientations. We can then apply this sample to what we call an electron microscopy grid. It's called a grid because it has a grid of different sections on it. Often these are made out of copper and gold, and they're quite small, maybe the size of your fingernail, or about 5 millimeters across in general. So we can take this sample, as you hopefully saw, so we can take the sample and we can apply this to a grid. We put a big blob of our sample on there, and then we use blotting paper to remove most of that sample, because we just want a very, very, very thin layer of liquid. We then freeze this inside liquid ethane in order to freeze it very quickly. And if we kind of zoom in and look on the side of what this grid looks like after it's been frozen, you can see here you have a support. So that was that kind of grid, in this case, even smaller parts of the grid, this whole carbon grid. And in between there, you have this very thin layer of your sample. And if we zoom in, you can see we have this thin layer. And within that layer, you would have your protein molecule in all these different orientations. So what you'll see in a second, what that means is if we can image this from above here, we'd be able to image your sample in a frozen layer of ice. So it's not quite in solution, but it's sort of flash frozen in solution. And hopefully we'd be able to see that molecule in different orientations. So we transfer that grid. This is a sample holder here, or an interpretation of that, in an electron microscope. And the electron microscope has a beam of electrons here. It's an electron gun that accelerates up at a particular voltage. And then it has a series of apertures and magnetic lenses here that allow it to magnify many times. The electron beams come down, they get magnified onto this sample, and then the image from that sample is magnified out and into a detector at the bottom. So really electron gun at the top. It's like a accelerator to get those electrons running very quickly to give them higher energy in one particular energy. And then a series of apertures and lenses that allow it to magnify to quite high magnification. So if you have like a light microscope, back in my university days, our microscopes only really went to about 100 times magnification. But in an electron microscope, we can go up to sort of 100,000 or 200,000 times magnification to create these images here. Now, these images are very poor, and that's just from how the electrons interact with the sample. And even over the internet here, I imagine they're even worse. But really, this is a picture of ice, this amorphous ice or vitreous ice. And then all these small little kind of speckles inside here are actually protein molecules. Those protein molecules we saw before in different orientation. So we're taking a projection or an image using that electron microscope to get all these small little protein molecules in a particular kind of different orientations. And then what Jochen Frank quite nicely showed is if we could take all these images and we just extract them out of our electron micrographs, and you should see some of them here have been shown with circles, triangles, pentagons, and squares, just to suggest they're slightly different orientations. If we take all of those and then we kind of are able to extract them out, what we'll be able to do is see this molecule in different orientations. So they're trying to show here with these circles that these orientations might be the same, this pentagon might be the same, this triangle might be the same, and this square might be the same. And if we only had 8 of these molecules, 8 of these images, we wouldn't be able to do much. But rather than just have 8 of them, what we actually do is take thousands of those images. And because there's multiple per image, we end up with usually hundreds of thousands. Oops, technology is not going well today. We usually end up with hundreds of thousands of these small particles, and we can average those together using this method of single particle analysis, just as if I were to take images of my face or my head from different orientations. I could build up a 3-dimensional image of what my head or my face looks like. Here we can take hundreds of thousands of these particles, we can average them together, and we can understand what a protein molecule looks like. And in that, we can usually build these atomic models where we have all these different atoms in different spaces. And really that 3-dimensional image of how all those atoms are arranged against one another allows us to hypothesize about how these proteins work. And we often test those using mutagenesis and other methods. So I think that's a small little introduction, but effectively we can use these electron microscopes to understand where all the atoms are in our particular protein of interest, as long as we can purify it and get it to an electron microscope. So my lab's been lucky enough to work on our own proteins and also work in collaboration with other people. And we're able to look at, vastly different types of molecules using this method of cryogenic microscopy. Some of them being ATP synthase, as you'll see today, these small molecules, SLC22s, transporters that James might be talking about, chaperonins, which are very large protein cages to do with folding we've worked on in the past. During the pandemic, like many other people, we're able to use electron microscopy to look at the spike protein on the outside of SARS-CoV-2, how it binds to different therapeutics like antibodies. We work closely with a lab just next door to us that works on potassium channels. We work with other people at other universities also to look at glutamate transporters and nucleosome core particles. And I guess what I'm trying to show with this image is we can look at things inside the membrane, we can look at soluble proteins, we can look at proteins with DNA attached, we can look at, proteins which are just one molecule, ones that might be tetramers or trimers, and really electron microscopy, unlike X-ray crystallography, has allowed us to really look at a whole different type of group of proteins than otherwise we had done before. But today, I'm really only going to talk for half an hour, so I'm just going to be talking about this molecule here, ATP synthase, which is what me and Francisco probably is our favorite protein, what we've kind of spent most of our careers on working on. And instead just trying to understand how this particular molecule works and how we're able to use cryo-electron microscopy to understand some of the catalysis that happens inside this molecule. So hopefully most of you have heard of what ATP is. Usually in school and university, we refer to it as the energy currency of the cell. And ATP is really this adenosine triphosphate. So you have this adenosine ring here, and attached to it, we have 3 phosphate groups on the end here, hence the tri for 3 phosphates. And this molecule can be hydrolyzed. So we just add a molecule of water, and that allows us to take the ATP to ADP. So the triphosphate down to diphosphate. So it splits this molecule in 2. And we get inorganic phosphate, and then we get adenosine, so adenosine ring, and then 2 of these phosphates. And this is quite an interesting reaction. It's an energetically favorable reaction in that the products here, the ADP and phosphate, have a lower delta G than the reactants here on the side. So generally in chemistry, we refer to this as an energetically favorable reaction, and the cell's quite clever. It can even get a bit more energy out of it by controlling the chemical environment in certain situations. So biology, again, is pretty clever. It can couple this hydrolysis, which is an energetically favorable reaction, and it can couple it to other energetically unfavorable reactions. Now, they might be conformational changes that you have. They might be polymerization, like DNA polymerization, for example, or it might be transport, sort of active transport. You can have these ATP-dependent transporters. So anything inside the cell that really, let's say, requires energy, or at least is energetically unfavorable, what biology can do is couple that energetically unfavorable reaction to just this hydrolysis of ATP. And that's really how the vast majority of molecular processes happen inside us as a human. And in fact, a human, we usually turn over our own body weight of this molecule in a 24-hour period. So the only problem with this is if we carry on hydrolyzing all these molecules, we'd run out of them inside our cells, and therefore all organisms really need to recycle this or synthesize this molecule. So I've only talked about hydrolysis, but what we also need to do is we need to synthesize this. We take adenosine diphosphate and phosphate, and we kind of do a condensation reaction, and then we end up with ATP on the end. Now, in most cells, most of the ATP is generated by this molecule here, which we refer to as F₁-ATP synthase, which is what I'm talking about today. We call it F1 just because actually Francisco was talking about regulation before. There's a factor called factor 1, which inhibits this motor up the top here called factor 1, and that binds in here, so we call it 1. And we call this bottom motor here FO, as you'll see in a second, because it binds to oligomycin, so that binds and blocks down here. So really the molecule is one complex together, but we often think about it, as you'll see in this talk, as either F1 or FO, because they can independently work separate to one another. And as you'll see, the FO is driven by protons, causing rotation down here, and F1 is driven or synthesizes this ATP molecule up here in a binding site. So what powers this? Well, again, in most cells, this could either be a mitochondria or it could be a bacteria. They set up a system where you have 2 different membranes. And inside of here, you have cofactors. You have a whole range of molecules I'm not going to talk about today. But really, these cofactors get made and pushed around, and this allows protons to travel from one side of the membrane to the other. So this would be a periplasm in a bacteria or an intermembrane space in a mitochondria. And these protons build up a proton motor force in this space in between these 2 membranes. And this proton motor force is what powers this rotary motor, this ATP synthase, by going through this motor. And then that gives them this, again, coming back to that energetically unfavorable reaction now, and that couples this energetically favorable movement of protons to the unfavorable reaction of going ADP and phosphate. We made a movie of that many years ago, which is this movie. You can see at the bottom here, we have all these protons, which are sort of at a high PMS. And one of these protons you'll see, they'll sequentially go into this rotor. They cause rotation of this central rotor here in the FO. And this rotation is transferred to movements in this F1 group up the top here, which cause these conformational changes. And that cyclic conformational changes, as you'll see later in my talk, what you have here is binding sites which change from your bind ADP phosphate to providing a chemical environment to make ATP. So we show that again. Oh, lost my mouse. You can see as we come along here, these protons rotate around here. As this rotation happens, you get conformational changes, you get binding of ADP and phosphate in, you change this chemical environment, and that allows you to create this ATP or synthesize that ATP. So hopefully that came across okay over Zoom. So today I'm going to be talking about a work that was done by Megda Sopti in my lab a few years ago. And to begin with, I've just been talking about this whole molecule list, FO and F1, but actually what we're just going to be talking about today for simplicity is we're just going to talk about the isolated F1-ATPase. So we talked about purifying proteins before. What we can do is just purify this F1-ATPase. We can still understand how that works. In this case, we're going to talk about it in a hydrolysis setting. So I gave a brief introduction of how synthesis occurs, but really during the 70s and 80s and 90s, this man, Paul Boyer, hypothesized how this molecule would work long before we had structures, just based on very clear, beautiful biochemical experiments. So he hypothesized how he did this with a kind of a schematic or a drawing here. And he's talked about synthesis here and hydrolysis, which are kind of interchangeable. We'll talk about that. So at least in synthesis, you could imagine you have those protons driving this process. They're kind of going across this membrane. In hydrolysis, those protons are actually getting pumped because the thing, if it's not kind of inhibited or looked after in any way, is able to work in hydrolysis as well. Although obviously in the cell, we generally want to work this in synthesis, but in a test tube, we can get it to work either way if we wanted to. So what he hypothesized, even without the structures, is this molecule has 3 binding sites. So what is shown here is this is all kind of the same identical binding site here. We usually show these subunits as yellow, as you'll see later in the talk, and these are catalytic active binding sites. And in the middle here, which in the video beforehand and in future, the gamma subunit, which we usually show as blue, is this sort of bit that was rotating round based on this proton motive force. So what he said was there were 3 different binding sites. You had one here, which had a low Km for ADP binding and phosphate binding. You had another one, which had a high Km, in this case here for ATP, where you'd be having your synthesis happening. And then you have another binding site here that would have a low Km for both of them. And what he said was ADP and phosphate would come in and bind to this tight binding site. Protons would then go through here and cause a rotation. They'd change the chemical environment in this binding site, and that would catalyze this ADP and phosphate ADP. You then get more rotation of this gamma subunit driven by the protons, and this binding site and this one, which would then go open, and you'd have exchange of ATP. ATP would come off, and then it would rotate around again in ATP. By cycling and going around, for every rotation, you would then get 3 molecules of ATP made. And this works in reverse. If you get hydrolysis, you imagine the opposite is true. You'd have a binding site here. We have low ATP, so ATP comes into this open binding site. We then get rotation in the gamma subunit. We then get a hydrolysis of ATP here. And then we get another site here, which doesn't have a very good binding affinity for ADP, but would be ADP. And then that rotates around. So again, for every rotation, we would use 3 molecules of ATP. And about 1994, this work was pretty much almost fully proved by Jan Peter Abrahams and John Walker's group. And what they saw here was 3 different binding sites. So again, I'm just talking about these yellow bits here because the red ones are catalytically inactive. If you look closely where my pointer is, you can sort of see they've got molecules here. This is an ADP in a binding site. This one's an ATP, and this one is empty or open. So they quite nicely showed by solving a crystal structure of the F1-ATPase, you'd have an empty site like they were suggesting. You'd have a site that likes binding ADP that they see here, and you'd have a site which has kind of undergone catalysis where you'd have an ATP. So by using this crystal structure, they could begin to understand hopefully exactly how this molecule works. Now, as well as using crystallography, a good friend of ours, Hiroaki Noji, was able to do a beautiful experiment with the F1-ATPase. and put a very large fluorescent active molecule on the F₁-ATPase. So, and fix this to, in this case, a glass cover slip. And if you observe this through a fluorescent microscope, because no one really believed at the time that it might rotate, what you could actually do is see the rotation of this gamma subunit relative to these alpha and beta subunits here by rotating around. And if you look on the right, here's a video of that, and you can actually fully see that you can see this rotation. And more than that, occasionally what you'll see is it dwells, where I've drawn these small triangles here, that it dwells at 120 degrees. Just like Boyer had said that you'd have these 120-degree rotations, because you have 3 different binding sites where it might dwell around. Now, unfortunately for science, though, it got a little bit more complicated than that. They redid this experiment using the F₁-ATPase here, and they conjugated a small B to it rather than a large filament. And what they could see when they looked at this bead now rotating rather than a very large actin filament is, rather than 120-degree rotations, just looking at the centroid of this bead here, at 120-degree rotation, we're split into 80 degrees, 40 degrees, 80 degrees, 40 degrees, and 80 degrees, 40 degrees. In other words, this molecule seems to work a bit more complicated than Boyer hypothesized, or John Walker had shown with that structure, that rather than having these kind of quite clear just 3 dwells, we actually have 6 dwells as we rotate around, and they could do a lot of statistics for that. And these single molecule groups, they actually went on and did dozens and dozens of papers to try and dissect exactly how that rotation happened. And what they came up with was what they call here this rotary scheme, rotary catalysis scheme, which is quite challenging to understand the first time you look at. But they're trying to simplify things. So what they have is, they have this rotation. You can see it 0 degrees, 80 degrees, 120, 200. These are just those steps that I showed you beforehand. And then at any particular place on this diagram, they're just talking about what would happen in this particular rotary state. And you can see that this blue thing, this gamma, it rotates by that 80 degrees. Yep. What they show here is these yellow subunits, which are these beta subunits. And then within these subunits, they're showing you what is happening in that binding site. So for example, we've already talked about that one site might be empty, one site might be ADP and phosphate band, and one site might be ATP. And they're always looking at in hydrolysis here, because they're just looking at the isolated F1. So we start here at position 1 or 0 degrees. The first thing that happens is we get this 80-degree rotation of the gamma subunit. And we also get binding of ATP and release of ATP. So you can see this particular site here, which is highlighted in bold, it's gone from empty and it's added ATP. And this site here, which isn't in bold because I don't talk about this yet, but it has ADP here, and this ADP is released. So what that really meant was they could try and dissect what happened in one particular catalytic site at every rotary state for this rotating gamma subunit. So we look here at this bold one, it would start out empty, it would bind ATP, and then nothing would happen. We'd rotate another sort of 40 degrees, it'd rotate another 80 degrees, and you get to this site here, a 200-degree rotation of the gamma subunit after you bound ATP, and this is where catalysis would occur. And then after that, you get a 40-degree rotation, you then get ADP release and phosphate release, and you come back. So really, there's just 2 interesting parts they care about. There's a place here where you're empty and you bind ATP, and there's an 80-degree rotation. They call this the ATP binding stroke. And then nothing really happens until about here, and then they call this the catalytic dwell because it's undergoing catalysis. And then you get a hydrolysis stroke here. So after here, after catalysis, you also get a hydrolysis stroke, and then you just get release of your products. So what they really cared about is like, how do you go from empty, your ATP binding dwell, you're waiting, to binding ATP, and how do you go from catalysis to over here? And because the molecule has 3 subunits in it, has 3-fold symmetry, there's really only 2 different structures we're going to look for here, or different states of this molecule. I've shown them here in green and magenta, but effectively all these green ones are the same, they're just rotated by 120 degrees, and all these pink ones are the same, they're just rotated by 120 degrees. And in the field, we already knew what these pink ones look like, this catalytic well. That was that crystal structure that I showed you that from John Walker's grad group from the mid-90s. But we really had no idea what the structure was of this green one, in this case, this ATP binding well. And that's really because when we look at the single molecule, it spends very little amount of time there. And when you run out of ATP, you end up dwelling in this catalytic well. So our historical methods of X-ray crystallography were never going to be allowing us to understand what this green one looked like, only this pink one. So we decided to use a mutant to try and understand that. This mutant's called a temperature-sensitive mutant. It was discovered by Yogi Noji and others. And what they were able to see is at 28 degrees, I won't go too much into this data because it's quite challenging again to understand because they've changed how to look at it. But if you just introduce this mutant, which is on this yellow subunit, this beta subunit, E190D, it's just a glutamate to an aspartate, you're able to make your molecule temperature sensitive. So at 28 degrees, your molecule generally bound in this catalytic well. But if we cooled that molecule down, it ended up dwelling in this binding well. So what that means was if we were to image this at 28 degrees, we would already get the structure which John Walker had already shown, this kind of catalytic well. Whereas if we image it at 10 degrees, we should be able to get this binding well or something that at least looks quite similar to this binding well. So high temperatures favor this catalytic well, which we already knew what it should look like. But low temperatures favor this new interesting binding well. So this is where cryo-EM could do something that X-ray crystallography wasn't able to do in the past. I showed you that video at the start. You'd have your molecule in solution, we can add it to a grid, and then we freeze that grid, and then we look at those proteins using electron microscope. But what we can do is we add this to a grid in a very temperature-controlled environment. So if we just take this mutant, this beta E190D mutant, if we add ATP, so it's rotating, and then we image it, we freeze it from either 28 degrees or 10 degrees, we should be able to weight our experiment or the structures that we solved either towards that catalytic well or the binding well. So we did this with the PCR machine, and then just to make sure everything was the right temperature, we added this magnesium ATP, applied it to a grid, froze it, and then we image it in electron microscope. So you can see here again, like in the introductory video, we have all these small little particles of what our molecule looks like. We can then extract out these small little particles and then solve structures either from 28 degrees or 10 degrees. So in 28 degrees, you can see here we can solve this structure of that catalytic well. This is akin to John Walker structures of the mid-90s. It shows the molecule kind of waiting for catalysis or dwelling around that catalysis. But then quite excitingly for us at 10 degrees, we're able to get this binding dwell or temperature sensitive dwell. So we can now see the molecule in this new conformation we hadn't seen before. And you can see on the left here, this is what our cryo-EM maps look like. The blue thing here is this gamma subunit. And you can see versus at 28 degrees to 10 degrees, it's rotated by about 44 degrees, which is what they'd expect. And you can see here in the density, we could work out what was an ATP molecule, an ADP molecule, where magnesiums were, and where certain different amino acids were. So now we have a whole new structure effectively to analyze. So we have two structures, this catalytic well, which again, like I said, was very similar to this crystal structure that was solved by John Walker, of like an open site that was bound at ADP, a closed site and a closed site. And we have a binding dwell structure, a new structure where this gamma subunit is rotated by about 40 degrees, giving us effectively 6 different binding sites. Because remember, this structure has 3 of these yellow subunits. This structure has 3. So we have 3 of these different beta sites. So we can track those, we can morph those in between what they look like. You'd have an open site, this then moves to a half closed site, and then moves to a fully closed site, which John Walker had seen before, a half open site, and opens up. And really this molecule is just moving around this, what I've shown here with a dotted line, is where you'd have your binding site of ATP or catalysis. it's really just changing that chemical environment to allow it to bind, in this case ATP, allow it to undergo hydrolysis in that section, and really just moving all the atoms around that particular interface. So what really changes there? Hopefully remember from the rotary catalysis, the first thing we cared about was going from 1 to 2. So this is where you get ATP binding. So we first of all get weak ATP binding, where we have these arginine fingers, which are quite separated from our phosphate. And then what happens when we get that ATP binding stroke is we go from a loose binding to a very tight binding. So these arginines here, arginine fingers, come down and lock in on the end of this ATP molecule here. Sorry, so this is the adenosine ring, and then 3 phosphates and a magnesium on the end. And then this is our anchoring kind of at the end here. So then not much happens, but then you remember we talked about hydrolysis. So then we wait until hydrolysis. Hydrolysis in our eyes looks very similar to this, but then after hydrolysis, we get movement of these arginine fingers. They slightly move up and they allow separation of that gamma phosphate. They come to a half open site, and then fully after we get hydrolysis, we open up to a fully open site, which then allows nucleotide exchange. So ADP can be released and phosphate can be released. So really what's happening is you're just getting this movement of these arginines are coming down and binding to this phosphate on the end. We're then waiting for hydrolysis, and then once hydrolysis occurs, we open up and the arginine fingers open up, and then we fully open to get to this site. And obviously we're just looking at one site, but this is coordinated at all the other sites at the same time through the gamma cycle. So again, we start with the catalytic well. Here what we get is release of ADP and phosphate. We then wait for catalysis. Once catalysis has occurred from magnesium ATP to ADP and phosphate, we get an opening of this beta subunit, which we call a hydrolysis stroke, which from single molecule, we know how much torque is generated from that stroke. We then get to the binding well, where we're waiting to bind ATP. So first of all, magnesium ATP comes in and binds weakly to one of these sites. And then as it transitions from this weak binding to a tight binding, going from a half closed to a closed, it moves down and clamps onto the ATP and causes rotation of this gamma subunit, and this gives us that ADP binding stroke, which gives out about 50 piconewton meters of torque. Hopefully this comes across again over the internet. Sometimes it doesn't work too well over the internet, but better in person. But we can think about this in terms of a movie of how these things move around. So this is the F₁-ATPase. We're viewing it, in this case, from the membrane. And this blue subunit's the gamma subunit. You can see there's already molecules of ADP and ATP bound in these sites. And then you can see a small white triangle here that we're pointing to a binding site here. So this is a zoom in of this particular binding site here. So on the right here, you're just looking at one site. So what we can then do is just play this movie through, which goes through this morphing, and it shows you this hydrolysis. And the first thing that happens is we get weak binding of ATP. So we call this loose binding of ATP, where these arginine fingers here haven't come down and bind. And then we get this full-on tight binding, or this binding stroke of ATP. So what happened there is these arginine fingers came down and clamped onto here. And on the rotation, we've got an 80-degree rotation or this binding stroke. So I'll just rewind that. You can see that on the right here, we're loose. And if you look on the left here this time, you see rotation as this speed. We then rotate a bit more, we go to a fully open site, phosphates released, ADP's released, and this just cycles around and around and around. And you can imagine that just goes and goes and goes and goes. And each time, each rotation, we're effectively taking an ATP, hydrolyzing it, we're releasing products, taking ATP in, at each side as they go around. So in summary, this is our F₁-ATPase work. We can see that it has 2 strokes, has an 80-degree ATP binding stroke, a 40-degree ATP hydrolysis stroke, and now we've seen these both in simple molecule and also in our cryo structural studies. These arginine fingers are crucial for binding and allowing that catalysis to happen. And we could also think we see a backdoor that allows independent binding of magnesium and phosphate. There's still an open question for us is actually how these sites are able to communicate to one another. None of our structures have really told us yet how each site effectively knows what the other side is doing. So for actually to allow rotation to happen, you don't just need one site working all the time like I show a zoom in. You actually need all 3 sites having coordination to, not rotate until hydrolysis occurs, not rotate until certain substrates are released. And that's something that we still try and think about and come up with experiments to understand. Just in short, most of this work was done by Magnus Opti in my lab. It was funded by the National Health and Medical Research Council. And all of our work on F₁-ATPase is done in collaboration with Hiroki Noji and Boshi Uno, who are at the University of Tokyo. And I'll be very happy to take any questions if anyone's keen. Yeah? Is that all right, Francisco? Yeah.
Well, first of all, after one amazing talk, I have never seen all the hydrolysis steps described in such detail. So thank you so much.
No worries.
I'm gonna switch over in Spanish to ask the students if they are willing to ask questions right now.
Okay.
Chicos, adelante, tenemos el doctor Alastair se tiene que ir ahorita en 15 minutos. Tenemos 15 minutos para aprovechar para hacer preguntas. Los estudiantes o cualquier persona que esté conectada adelante puede aprovechar para preguntar lo que guste. Ya sea pueden preguntar en español y lo pueden preguntar o lo pueden escribir sin problema, yo lo traduzco, o pueden incluso ustedes preguntar en inglés sin ningún problema. Tenemos el micrófono abierto para preguntas. Adelante, Fidel.
Hi, Dr. Alastair. First, I want to apologize for my English. I'm not the best speaker. So I have 2 questions. First, which organisms have you been working in this project? I don't know, E. coli or mitochondrial F1?
So I didn't go too much into that. We actually work on a whole different number of ATP synthetases. But today I actually spoke about this molecule here. So it's from a bacillus species that we often refer to as PS3. And the reason why we did that is it was identified in the 80s. It became a model organism. So, for example, here, Yoshida's lab and Kinoshita, they'd always used this molecule of F₁-ATPase, which was from a bacteria called bacillus PS3. But in our lab, we worked on yeast, human, bovine, E. coli, Pseudomonas, just recently F₁-ATPase phase. So we I didn't go into it today, but we often chop and change to answer specific questions. But in this case, because we were interested in rotation and substepping, we used bacillus PS3.
Okay, and another question. In all the organisms that you have been working with, have you ever found some difference in the rates of rotation?
Oh, yeah. There is a huge difference in the rates of rotation. It operates at about, I think it's about 6,000 hertz. Is that right? Yeah, about 6,000 hertz. In other words, it gets rotation about. But they're able to get up to about, I think, tens of thousands of rotations per minute. So they're much higher. Humans a bit slower, at least in our hand. yeah, there's a huge range in different speeds of rotation. But a lot of this is probably just due to the particular temperature that they grow at. But if it's being connected, they can get away with temperature. I didn't go into it, but that was kind of the one main thing that I skipped. You can see here, you're looking at this is the 75 rotations in the system at the 10 degrees, much lower. Is it down by the water tube? And that's why one of the reasons why we used it. So this
Thank you.
Thanks.
Okay, Alastair. Professor Garcia says thanks a lot and cheers from all the way to Nam, Mexico. And we have two questions. One is from James. James, go ahead.
Oh, I had a quick question, specific question, Alastair.
Yeah.
In hydrolysis mode, the water molecule responsible for hydrolysis, is that bound before ATP is bound? Is that coordinated by the protein before ATP comes in? Or does it come in after the hydrolysis stroke? Is that right?
So the water is always there. So I'm not quite sure you can see it on the slide. Probably isn't the best diagram for it here. I'm not quite sure. I left this in because it was from a different talk, actually. This is the water at the back. I'm not much showing. Yeah, so just at the back here, which is held by that glutamate. So that we believe the water is always there, even before you get the hydrolysis stroke. But I don't have a good explanation for this because it's not like the water is always there, but it has to overcome a particular entropy to allow you to undergo that hydrolysis, and that's basically based on distance. So it's always going to be there, but effectively it's like a timing thing. And actually, related to the previous question, we think about these things being very fast because there might be 6,000 times a minute. But if you think about your car engine, it's about, your car engines go at about 6,000 times a minute. That's extremely slow on the biological scale. So we think a lot of that is due to this effectively catalysis and how slow that is, because you've got to, usually when we look at these molecules, they're limited by catalysis. You're basically stored there, you've got a water next to your gamma phosphate, and somehow you have to go through a planar system where effectively your phosphate turns planar and comes off the other side. And that appears to be a timing situation, which is why partly rotation is so dependent on temperature. So it's just waiting its timing, and then a particular event that happens that allows that hydrolysis occurs. But compared to other molecules, this one is incredibly slow. Many, many orders of magnitude slower than a proper enzyme like lysozyme or something.
Thanks.
Yeah.
Okay, so we have a question from Gilberto. So go ahead, Gilberto.
Oh, hi.
Hi there.
I was just thinking about how, well, first of all, thanks for the great talk. It was amazing.
Thanks for coming.
I was thinking about how it's possible that a specific point mutation on the beta subunit helps or promotes the binding dual over the dual. Can you explain us a little bit more about that?
We don't know that answer. I guess I partly alluded to it in the answer to James's question that this E190D, oops, I'm trying to scroll on that. Here we go. The place of the mutation is this residue at the top here. So that residue is coordinating the water that allows the hydrolysis to happen. So we believe, and it does, it slows down catalysis because it's holding that water a little bit further away from where that is. But we don't understand yet why it's causing the binding dwell to be much, much, much longer. Partly because we can't see waters in the structures that I've properly shown you today. We have that here. So our resolution of these structures is good, but for example, we can see the magnesium, but we can't see where that water is. But it's probably something to do with how this ATP is binding. So as well as being due to being involved in catalysis, that water is probably helping that binding happening. Or it's something to do with how that information I came back to is being coordinated from your different sides. But we don't really know. I guess we've got proper answer. I don't know what's happening. Because it is just, all we're doing is removing one carbon. Yeah, we're going from a glutamate to an aspartate.
Okay, and we have another question by Oscar. His question is, what advantages do you guys have by using 3D structures using this method of cryonium opposed to use X-ray crystallography?
So I guess our big one is, I mean, a fair bit of what we've tried to do, and well, we'll go back here even. The first one is, during my whole PhD, we were devoted to try and understand how structures operate inside the membrane. And crystallography was very challenging to do that. Not impossible, but we used to spend years setting up these crystal trays to try and coax these molecules to form crystals that would normally be in a membrane. And that was very, very challenging because this membrane region usually didn't want to form what we call crystal contacts. A crystal is sort of like this array of how they put together. So the reason why we originally went into cryo-EM wasn't to kind of understand dynamics, which is more what actually came out of it, but because we worked on these membrane proteins and none of them really worked in crystallography. It was just too challenging, too impossible. Now, the other thing that came out of it is what we realized quite quickly is usually in crystallography, we set up these trays like overnight or even for weeks or months. And therefore, we're just looking at one specific static structure. And it was great. I mean, John Walker's work on this F1 was kind of game-changing at the time. Obviously, he won a Nobel Prize for it. And it was extremely challenging to even solve this structure due to its size, how poor the crystal was, everything else. But you're only ever really able to get one particular confirmation for it. They did, spent 20 years, they were able to change a lot of conditions around here and saw small sub-rotations. But these large rotations or large changes, they weren't able to see using crystallography. So by using cryo-EM, we were able to, as I kind of suggested, we were able to manipulate this environment, to weight that environment, to understand how it would be in different settings. So I guess what I wanted to do when I started my group was to try and marry some of this single molecule work with structural biology. And in hindsight, crystallography was never going to be able to do that because we just couldn't get it in that setting. So we spent decades trying that, whereas I guess cryo can do that. And NMR can do that as well. It's just different, very challenging.
These proteins are too big, no?
But yeah, they would be too big. I don't know what your friends would think, but too big, but you can still supposedly understand some dynamics quite well with NMR. You can certainly get an idea of flexibility and these other things, but it would, wouldn't do too well with an F₁-ATPase in two different conformations.
Okay, Professor Alastair. So we know you have another appointment.
I do, yes.
We're going to stop the questions here. We would like to show you that we have a certificate
thank you.
For your talk. I'll send it to your email.
Yeah.
1 second. So I'll send it to your email, and thank you so much for your great talk. It's a great honor. It's the first time you're doing a talk in Mexico, so hopefully maybe in next year or later on we can invite you to give a talk and show you around.
Yeah.
So
thanks so much for having me and hosting me. And great to see you again.
Okay. So
Okay.
let's keep in touch and have a great afternoon. And we can start with James.
Yep.
Okay.
See you soon.
Thank you so much.
Best of luck, James.
Tough act to follow. All right.
You have great data, James. Do you want to start by before we start? I'm going to give like a brief introduction of yours in Spanish. I don't know if you want to try to we can make a small test to see if we can connect your your slides.
Cool. Can you see them?
Yes.
Great.
I'm going to stop the recording so that I can make one recording and then the other one separate. Is it okay by you if we record your talk?
Yeah, totally.
Okay. All right. So I'll start to give your introduction in Spanish, and then I'll switch to English, okay? Buenas tardes, estudiantes y otros compañeros docentes y público en general. Nuestro siguiente seminario lo impartirá el investigador James Davies. Él estudió la licenciatura en la Universidad de Canterbury en Nueva Zelanda. Él se graduó en el 2021. Realizó una estancia postdoctoral en la misma universidad, estudió el doctorado y realizó una estancia postdoctoral en la Universidad de Estocolmo, Suecia, del 2022 al 2024. Actualmente está realizando una estancia postdoctoral en el laboratorio del Dr. Alastair Stewart en el Dr. Chang Cardiac Research Institute, que se encuentra en Sydney, Australia. Ha publicado más de 2 artículos internacionales de investigación y tiene más de 200 citas. Él es un biólogo estructural que se enfoca en la estructura de los transportadores de sodio y le agrada mucho la parte computacional de la ciencia, por lo que es una excelente combinación. Es una de las personas que he visto que obtienen modelos estructurales súper suaves, justo porque sabe cómo explotar los distintos algoritmos y programas de modelaje y los puede aplicar con los datos estructurales que lo obtiene. Las dinámicas moleculares que hace están súper fáciles. Okay, so our next seminar will be presented by soon-to-be Professor James Davies. He completed his undergraduate degree at the University of Canterbury in New Zealand. He graduated, he also did his PhD over there. He graduated in 2021. He then completed a postdoctoral fellowship at Stockholm University in Sweden from 2022 to 2024. He's currently doing a postdoctoral fellowship in Dr. Alastair Stewart's laboratory at the Dr. Chang Cardiac Research Institute in Sydney, Australia. He has published more than 12 international research articles with over 200 citations. He's a structural biologist focusing on the structure of sodium transporters. He also enjoys the computational side of science, which makes for an excellent combination for his work. He coined the term elevator with an operator for the mechanism of transporters. So for me, it's a great honor to present to you all the one and only from Christchurch, New Zealand, James Davies.
Thank you, Francisco, for the very kind introduction. And thanks everyone for turning up today. I wanted to talk about sort of 2 case studies of cryo-EM being able to allow us to solve the structure of small membrane proteins. So I work on transporters, as Francisco was saying. These proteins are about 1/10 the size of ATP synthase that Francisco and Alastair work on. So they present this sort of special challenge or extra challenge for cryo-EM. So they're right on the limit of what we can do with the microscope. So today I'm going to talk about a transporter from humans called Oxygen-2. It's a carnitine transporter, and it uses sodium to sort of power this transport. And then in the latter half, I'm going to talk about work during my PhD on a substrate binding protein dependent transporter called a trap transporter. And these are found in bacteria, they're not found in humans. So what a transporter is doing in the cell, just a little bit of background in case you didn't already know. One of the main jobs that they're responsible for is moving solutes and substrates that are low in concentration outside of the cell into the cell. And this is an energetically uphill or unfavorable process. So these proteins couple this uphill process to a downhill process, either ions moving down their electrochemical gradient, or we can use the ATP generated by ATP synthase to sort of, the hydrolysis of ATP, I should say, to power this movement into the cell. So we call this active transport, and I'm particularly focused on secondary active transport. So these transporter proteins undergo conformational changes to move the molecule into the cell across the membrane, but we all sort of use this main mechanism called alternating access to describe how they do that. And that's when you have these sort of, what we've got depicted here are bundles within the protein that sort of change and cycle conformations to have alternating access on either side of the membrane. So here it's outward open, so a substrate can come and bind in there. A little gate closes over there, occluding, we call this occlusion, and it cycles to this sort of airlock state here called occluded, where the protein is shut off essentially from both sides of the membrane. We then go to an inward state, and the protein can drop off the molecule on the other side into the cell. So we call this one the rocket switch alternating access mechanism. There are lots of other ones and lots of variations of this. Another one that we look at sometimes is the elevator movement, which is where you have this bundle here sort of moving like an elevator up and down in the membrane, taking substrate and that sort of driving ion down into the cell. So as you go around here, you can see that this bundle moves from this up state to this down state, where it can release substrate on the other side. So I guess nature's evolved to these proteins to use really different mechanisms to move substrates across the membrane. So the one that I've been focusing on in Alastair's lab is a carnitine transporter. It's a human transporter. You've got it in almost all of your cells. And its job is to move this molecule called carnitine here into the cell. And carnitine's job when it's in the cell is to help shuttle fatty acids into the mitochondria, where they're broken down by beta oxidation. You might remember from biochemistry 101. And then we can make acetyl-CoA and feed it into the citric acid cycle. So this transporter's got a pretty important job in making sure that the cell's got a continuous supply of carnitine. And to do this, it couples the movement against the carnitine gradient. So we've got 1 to 5 millimolar inside the cell, and micromolar outside of the cell. So it couples this process, which is really unfavorable, to the movement of sodium down its concentration gradient. You can see just here. So as I was saying, this protein is found throughout your body. If we look at mRNA sequence analysis, you can see here that it's got really high expression in the muscle, in the kidneys, in the heart, and the brain. So it's doing a really important job. If you have things going wrong with this transporter protein, it causes an inborn error of metabolism called systemic primary carnitine deficiency, or SPCD. It's really rare, but it's really serious because when you have problems with carnitine uptake, you can't do your beta oxidation. You get fatty acids beginning to accumulate throughout the body. And this can lead to all sorts of problems, but mainly heart failure. Very luckily, with early diagnosis and supplementation of carnitine, you can fix this problem. But there's lots of variants in this protein, or lots of mutations that affect the protein that we don't really know how they work or how they affect transport. And diagnosis can be quite difficult as well. So in the lab, my job is to make the protein and try to solve the structure and figure out how it works and how these mutants affect the transport function. So I make the protein. It's a human protein, so I want to make it in human cells to sort of make the most native form of it. So I use this method called the backmam system. I make what's called a backmid, which has got my gene for opt-in 2. I put a GFP tag on it, green fluorescent protein. We make this virus, baculovirus, in insect cells, and then we use this virus to infect human embryonic kidney cells, which has got all the instructions to make the protein. This process takes, I don't know, a couple of months, essentially, to go from plasmid to expressing protein in the cells. So once we've done that, I use traditional protein purification techniques to isolate the protein. I get the membranes of the cells. I use a nanobody, which is like an antibody, to sort of selectively bind this GFP. I cut this off here, and then I put it down a size exclusion column. Very similar to what Francisco does in his lab, but I guess the special bit here is that we're doing it in human cells. So with that protein, you can see here on the F-ATPase gel, it's relatively pure. This sort of smearing is because there's some glycosylation on the protein. We take that protein and we go to the microscope, as Alastair was showing. So we first start the sort of pipeline that we follow in Alastair's lab is we use our microscope, 200 kilovolt Talos Artica, to sort of screen conditions that are amenable for protein structure determination. So it wasn't quite as simple as it looked in that video that you've seen before. You have to work quite hard to get really nice distribution of the protein particles in the ice. So once we've got that, we go to a more powerful microscope down the coast here in Sydney. It's 300 kilovolts, and we collect really massive data sets. So we're not actually taking images of the protein, we're taking movies, and we average these movies to sort of get really good data of these particles. We have lots of computation that you then do on these micrographs. And this here is this kind of like the shapes that were in Alice's video, those different poses of the particle. This is called a 2D classification of all these transporter particles. And this here is this detergent micelle. So we solubilize these are membrane proteins, remember that, in a lipid environment. So we need to use a detergent to extract them, and we can see that in the image there. It's the micelle, it's the protein, and that's part of the protein outside it. So yeah, from 20,000 movies, we have 6.4 million pick particles, and we sort of refine them and whittle them down to a subset of really good signal to noise particles, which we can then use in a 3D reconstruction. So hopefully this plays. Here we have, just turn the, there we go. This is the, I'm not sure how well this is coming out, but this is a video of, I guess, the first structure of opt-in 2. You can see here, I've put in blue and orange, these are the two bundles of the transporter within the membrane. We have this green extracellular domain and a purple domain down here we call intracellular helix. And in gray, you can sort of see around the periphery of the protein, we've got these lipids and perhaps detergent molecules. But the really cool thing here is, well, first we solved a structure. This hasn't been done before. People have been waiting sort of 30 years for the structure. But we also solved it in an intermediate state. So if you remember back to those cycles I was showing you earlier, it goes through this sort of outward open, occluded, and then inward open state. So this is an occluded state or an intermediate state. You might think of it like a transition state in an enzyme. So in here, we have carnitine bound, which you can see a representation of electron density over here, but the gates that sort of close off the protein on the outside and on the inside are both closed. So this is a really valuable structure in that we can see how this protein binds the molecule that it transports, and also how it binds the ion that it co-transports. So here you can see a sort of representation of the structure in the ribbon format. If we take a cut through of the surface of the protein, we can see that there's these two little cavities here, one containing carnitine and one containing sodium. And if we look even closer at the electron density or the electrostatic potential map, we can see sodium down here coordinated to a water molecule, And up this cavity, we have water molecules within the protein, and we have carnitine over here. So there's two sort of separate, distinct sites where the protein is binding the things that it is transporting. So this is a view looking down at the protein, I guess, from outside the cell at the binding site. We can see that it's coordinated by amino acid residues on the positive charge side of carnitine, and on the negative charge side of carnitine, there's a salt bridge holding it there, and we have the sodium side over here. So sodium's bound kind of as we expected by oxygens of these residues. Carnitine, it was really interesting to see, forms a really strong salt bridge, and then there's an aromatic cage forming these things we call cation-pi interactions. So this is a positive charge. These are all pi electron-rich residues, and it's sort of forming an interaction here. So it's really held tightly on both sides of the molecule. This is that same view in 2 dimensions. So the carnitine binding site here and the sodium binding site here. And if we look at variants associated with the disease I was talking about, SBCD, we can see that very many of them localize to these sites, so to the carnitine binding site and the sodium binding site. And we know for some of these mutants that cause the disease, that they don't affect how the protein folds or the stability. The protein still gets to the membrane in your body, but it's we think that these mutations are actually affecting how they're binding carnitine and sodium. So we're starting to look at how the protein structure changes in the disease state. So to sort of follow that up, we're using another technique where we're making the transporter in these things called oocytes. They're frog egg cells, and we do 2-electrode voltage clamp electrophysiology. This is where we can inject current using microelectrodes and record the current as well, and keep the voltage clamped and sort of measure how the transporter is transporting the molecule. So in the presence of sodium, perhaps expectedly, we get nice transport. This graph means that there's transport here with sodium and not with cesium. So we could say transport is dependent on a sodium gradient. And as we sort of reduce that sodium gradient, the amount of sodium outside the oocyte, we get a reduction in the carnitine affinity. So this is carnitine along this axis. And as you go this way, you reduce sodium. And this midpoint here is the Michaelis constant, or the Km for carnitine. So reducing sodium, or the gradient, reduces the affinity to the affinity for carnitine. So altogether, I guess you could say that this is not really unexpected. Sodium is driving transport. And this Km here that we measured is quite high. So it's quite a high affinity transporter, around about 10 micromolar. So if we look at our structure and we go, these are the residues that are coordinating sodium, and those are the residues that are coordinating carnitine, we mutate them, and then do that same experiment where we titrated in carnitine to measure the rate of transport. We can see that mutating these residues, so asparagine 210 here, coordinating sodium, and N32 as well, that we're shifting that Km. So we're reducing the affinity for carnitine by mucking up the sodium site, essentially. So yeah, 20-fold carnitine Km increased here, 11-fold here. And if we mutate these residues here, you notice they're not on the graph because there was no current measured in the electrophysiology experiments. So these residues are all really important for the transport. So this is a little bit of a detailed site, but we've started to map how these sites might be connected together and how sort of allostery connects these sites, which is still a little bit tricky for us to work out. But we've sort of identified that this residue here, N32, which is forming a bond with the sodium ion and a hydrogen bonding interaction with the glutamine, is sort of this pathway back to this carnitine site. And really interestingly, we noticed that in a place called the Faroe Islands, SBCD has a prevalence of 1 in 300. And if you have a mutation that changes this asparagine to a serine, the disease state is associated with severe arrhythmia and sudden death. So we know that this bit is probably pretty important for how the transporter functions. So if you remember that cycle, we got the occluded state, but that's not enough information to tell us how it's working. So we also solved the inward-facing state, which is here. You can see here that these two bundles in the membrane have come apart, they've opened up. So this is what you might think of as the substrate and sodium release state. I'm not going to dwell too much on these detailed residues here, but if you kind of look over here, you can see a superposition of the structural change between the occluded and the inward state. So in the occluded state, in orange here, it's all tucked up against this blue bundle, whereas in the inward state, it's rotated about there, the gate's closed on top, and it's all opened up. And these residues that are sort of responsible for gating or closing the transporter off from either side, we see that they're actually involved in important interactions, but not only that, they're also positions where if you have mutations, you can get the disease. So altogether, we've sort of started to map for the first time how this transporter works. We've solved the structure here, the occluded loaded state. We've solved the structure here as well, but you can see here that we've got a lot more structural work to do to fully understand this transporter. So that's the transporter that's really important in humans. That's been the last, I guess, 2 years of my life, every day. But I wanted to talk about, I guess, my pet project, the one I did my PhD on, which is they're called trap transporters. And they're found in bacteria and archaea, but more commonly in bacteria. And they're not found in humans. I guess that's important because, I mean, they're interesting to study and think about how they work. But that means that potentially they could be antimicrobial target because they perform some really important roles in how bacteria interact with their environment. So traps, these are 3-part systems rather than this 1-part system I was talking about before into. So you have 2 parts in the membrane, M and Q, and you have this binding protein up here that sort of captures a molecule and delivers it to the membrane. These can be all sorts of molecules, mainly those with a negative charge group. So there's a carboxyl here, CO2-, or a sulfonate group. We're particularly interested in this molecule you've probably heard of called sialic acid. It's a sugar that's really commonly found in all of our cells as well. So yeah, as you see here, on the surface of our cells, we have these things called glycoproteins and glycosphingolipids, and at the end of these glycans, there's a sialic acid molecule. And what can happen is that the sialic acid molecule can be cleaved off, and then bacteria can uptake through these dedicated transporter systems of sialic acid, and then use it either as a source of energy, or they can do this really clever thing where they repurpose the sialic acid and put that sialic acid back onto the outside of their cells in a way of sort of mimicking or camouflaging as a human cell. So this one here, Haemophilus influenzae, is a really bad pathogen. Has a very complicated machinery in order to do this. So these sialidases, they sort of cleave off the terminal sialic acid here. And then, of course, being gram-negative bacteria, it has 2 membranes. So the sialic acid has to come through this outer membrane porin via passive diffusion. But then we have the system that I'm sort of interested in, where the substrate binding protein is sucked up by this sialic acid, is sucked up by this protein, and this protein then delivers it to the system here. And it's sort of like in the human case, we have a sodium gradient where that the protein uses to sort of drive this unfavorable process. So once sialic acid gets into the cell, as I was saying before, it can come this way to a synthetase and then be repurposed back onto the surface, or it can go through all these enzymes here and be fed into the citric acid cycle, cell wall biosynthesis. It can make ammonia, or it goes into glycolysis there. So a really important part of this process is that this is a way bacteria can camouflage themselves from our immune response. So when they do this, they look like our cells. So it sort of attenuates the immune response. So I guess, again, we turned to cryo-EM. I spent the first 2 or 3, actually 4 years of my PhD trying to crystallize this protein, and we got great crystals. I would fly over from New Zealand to Australia almost every month, take them to a synchrotron, which is a big particle accelerator, and shoot the crystals with X-rays, and they would never diffract. So it was a lot of wasted time and effort. And as I was sort of finishing my PhD, the cryo-EM resolution revolution started to happen, and we sort of quickly changed strategy. So I moved away from crystallography and tried some cryo-EM. But because this protein is really small and doesn't have anything outside the membrane, apart from the substrate binding protein, I guess, we knew we needed to do some protein engineering or use a different strategy in order to solve the structure. And that was to make this thing called a nanobody, a small antibody fragment. And I did some engineering to sort of fuse the scaffold protein onto this nanobody so that it became even bigger. So this nanobody we raised in yeast against this bacterial transporter. So it has really high binding affinity. It comes over here, and it sort of acts as a marker to sort of increase the size of the particle for cryo-EM, but also to help during this particle alignment process where we make that sort of 2D classification of the particles. So this is from a model organism, Photobacterium profundum. It's found in the ocean. It's not really a pathogen, but we picked this because it's a pressure-loving organism, and it's, I guess, potentially when we're doing crystallography, we screen proteins from many different bacteria for stability, and this sort of came across as a very stable version of the trap transporter. So this is the one we chose for cryo-EM studies. So I guess we solved different structures. We use different kinds of detergents. This is actually an amphipole, and this here is what we call a nanodisc. This is where I take lipid molecules and a belt protein, and I sort of insert this transporter into this nanodisc, which is supposed to be a more native state for structure determination, which was, yeah, it was really nice to solve this structure to a high resolution. I guess this is also one of the first times that this protein has been solved. So when we look at it in depth, we can see that it was in this inward-facing state, and that this transporter is actually an elevator. So as opposed to that rocker switch sort of mechanism I showed first, this is a protein that can move these bundles up and down through the membrane. And over here you can see these bundles in orange and yellow. This is the bit that moves up and down, and it's sort of cradled by these what I call arms here, these bits in pink, and this sort of moves up and down, well, potentially, against the thing we call a scaffold domain. So there's a scaffold and a transport domain. And this was a new finding for this, this group of transporters. So if we look at cutaway, we can see the substrate binding site. But critically here, we can also see the sodium binding sites as well. And we were lucky enough to see pretty decent density for sodium at these two sites here. And I guess in contrast to that octane-2 transporter, the sodiums are right here next to where the substrate binding site is. So they're not in this sort of allosteric site. So they kind of form part of the substrate binding site. So to test these sites, we did proteoliposome assays using radio-labeled sialic acid and the P subunit. And I guess an important bit to say here is that we tried to solve the structure with the P subunit, but when we measure it in the lab, these two things don't really interact that strongly. So they have a really low affinity. And we think that's to do with transport, so that this thing can come on and off the transporter. But it really didn't help us when we added this into the cryo-EM condition. So anyway, we purify this separately and then add it into an assay. This is a proteoliposome where we've embedded the transporter in here. And we sort of titrate in this P subunit, as well as we have a sodium gradient, and we measure the rate of transport. So we see max transport with sodium gradient, as you'd expect. but also a membrane potential. And this is mediated by, we use this thing called valinomycin. It's an ionophore. So we add this in and it moves potassium out. It creates a membrane potential. And that shows us that transport is electrogenic, which is not surprising, as we have a negative charge on the sialic acid. And we have two sites that bind sodium. So there's going to be a net movement of positive into this proteolipase. Transport requires the substrate binding protein. Also, not that surprising, but as you increase the amount of binding protein, you increase the rate of transport. And then when we titrate sodium or increase the sodium gradient outside, like we did with octane 2, we can measure the rate of transport and determine the Hill coefficient, which is greater than 2, which means at least 2 sodium ions are coming along for the ride, along with sialic acid. So there's obviously 2 bits to this transporter. We solved a cryo-EM structure without the substrate, even though we put the substrate in. So it seems to us that there's really low affinity of the transporter itself for the substrate. It's the complete opposite for the substrate binding protein. So this is some crystallography that I did. Alastair was briefly talking about, I think. I grew these crystals of this protein, purified protein, and I added in the substrate, sialic acid. And then we took it to the synchrotron, got some mass diffraction. We're very lucky to solve it to 1 angstrom resolution. This resolution, I can see water molecules, I can see hydrogens essentially on the side chains, which you can kind of see here. Here's the sialic acid, here's the water molecules all around it, helping coordinate it to the protein, and a bunch of really strong interactions like the salt bridge here between this negative charge and this arginine. When we do this technique in the lab to measure affinity, we can see it's really high affinity, 500 nanomolar. This is isothermal titration calorimetry. So I guess what we're showing here is that this binding protein really tightly binds sialic acid, which was known in the literature before we started this project, but we wanted to show it with this system. What we also showed, I guess this is another X-ray technique, and there's a lot going on here, but what I wanted to show was that when this is small angle X-ray scattering, so we have the protein in solution, we hit it with a beam of X-rays, and we measure that scatter, and then we look at our structures, our crystal structures, and we say, does that fit the small angle X-ray scattering in solution? So it's a way of sort of correlating your crystallography to what's actually happening in the tube. And what we got was that when you don't have that ligand in the aqueous state, you have the protein in an open state. You can see here the binding proteins sort of come apart. And actually, from our molecular dynamics simulations, the protein's really open in solution. So it's there waiting, I guess, ready for sialic acid. When you add sialic acid, it closes up and becomes more compact like this. So this is the state that we presume is the main interacting state with the transporter, and it's not stationed at the membrane. Sort of sialic acid comes along. It's this state that sort of diffuses around in the periplasm and comes to the transporter. So you can kind of see this conformational change in this movie here. Hopefully it plays. Oopsies. Yeah. There we go. So it's quite a dramatic change, and you have this bending of this hinge helix here around this ligand, and you have all these water molecules encased in it. So the binding is really complicated, and the protein becomes much more compact as it binds. So we can solve the crystal structure of the binding protein, we can solve the cryo-EM structure of the transporter, but what we couldn't do, which we really want to do, is see something like an occluded state or a transition state, where the binding protein is docked to this membrane protein, this transporter here. So we turn to computational methods. We use AlphaFold, which I'm sure you've all heard of for protein structure prediction. And we predicted, I guess, a number of different complexes. But this is the one I thought looked best, because I looked at another thing called coevolution. So I looked at the coevolution strength of residues between these two proteins, and then sort of mapped between them where I thought they would interact. And you can kind of see in this, if we open the structure up here like a book, You can see that I've sort of designated these places here where I think substrate binding protein sort of docks onto the transporter to hand off sialic acid. So that's just a prediction. It's not an experimental measurement. We wanted to test this in the lab. So this is very detailed, but from this model that I made, I guess I could see all these interactions, specifically what I thought were close to being interactions between the two proteins. So what I did was mutate them and then measure the transport activity in that proteoliposome assay that I would show. It turns out a number of these mutations at these hotspots, which is what I call them, they cause a significant reduction in the transport activity. So they don't stop the binding protein from being able to bind its substrate, but they do stop the actual rate of transport through the system. We also mutated a bunch of sites at the sodium and the substrate binding site to sort of validate them as well. So we came up with this mechanism, which Francisco mentioned, the elevator with an operator mechanism. We start off with substrate binding protein open, bind substrate. I call this the operator of the elevator. It's a bit cheesy in hindsight, but there it is. Once it's bound, it comes down to the transporter. It induces this upward motion of these orange and yellow bundles. Come up, sodium binds, sialic acid binds, the substrate binding protein comes off. The elevator then moves downward through the membrane. So you can see the substrate's moving downward, and then it leaves the transporter on the cytoplasmic side. The substrate binding protein being completely disengaged. So it's kind of reset to this downward state, and it's gone up like that. So that's just a model or for the mechanism. We're still trying to solve intermediate structures. We really want to solve this one here to sort of prove that there's this bundle, or at least measure how far up this bundle is traveling through the membrane. I've modeled it on the side there. This is a sort of 3D version of what I just showed you. But what you can see here, this is the sort of modeling I've been doing. This is the cryo-EM structure on the bottom, the crystal structure that I've put together. The substrate binding sites here. Here's the model I made using AlphaFold. The substrate binding site moves up, and you can see here that the distance is greatly reduced between these two sites. So how do you open a protein like this that is bound so tightly? So 500 nanomolars means it's not really opening up to release this. So I've identified this region here. I call it a latch, and it sort of extends from the surface of the protein via a water network back to the ligand. And when I mutate parts of this latch region, I can still bind sialic acid up here, but in that transport assay with the proteoliposomes, I have much greatly reduced transport. And when I mutate regions that interact with this latch in my models, I can see that I also reduce the rate of transport. So this is a sort of area that I'm exploring now as a way that this might allosterically cause this to open, and for this hinge to open up, and for this this ligand to be released. But this is really tentative modeling at the moment. So we've been doing lots of other work on traps from different systems, different bacteria. We work on pathogenic bacteria, Haemophilus. We've solved structures, and we've actually seen a dimer, really loose dimer, which we're unsure of whether it's biologically relevant, because we also saw this sort of alternate dimer or anti-parallel dimer where this is rotated 180 degrees upwards. So kind of we got really excited when we saw this, because we thought we had a down state and an up state, but it turns out that it's just rotated about 180 degrees. So it might be an artifact of how we purify the protein. And this structure here, we saw the sodium sites again. We had an empty substrate binding site. So it was really confirming what we've been thinking with the other system. And that is that this transporter has really low affinity, and it needs a substrate binding protein to come and deliver the substrate. So just recently, in the last week or so, I've actually had this paper accepted. We solved a structure where there was ligand in the binding site. And this is where I had to put this at, or my student had to put it at 30 millimolar to even see it. But this is another system from a really wacky bacteria that was found at the bottom of an oil well. But we're sort of doing this because we want to explore different traps and different bacteria to see if they all work the same way. So I guess in summary, I've told you about two different systems today. One's a human system, uses this rocker switch mechanism to move molecules across the membrane. It has micromolar affinity for its substrate carnitine. It's metabolically regulated. Has this unique sort of sodium allosteric binding site, which we don't really know how it couples to the carnitine, but it's definitely unique. But overall, we can sort of see that the system has evolved to do a job that's mostly homeostasis. I guess in contrast to that, we have bacterial system, this trap system here, where we have nanomolar affinity up here, really low affinity in the membrane. So all this affinity and specificity is actually outsourced to the P subunit. So this system seems to have evolved to function as a scavenging system so that the bacteria can take molecules that are really low in abundance outside of the cell and move them into the cell. So just to wrap up, I guess say thank you to everyone involved in the work. It's a really big team. Alastair and Jack on Opt-in 2. This kind of came from a little bit from Jack's PhD thesis. And we did the electrophysiology with Renee at the University of Sydney. And for the trap work, as a really huge team, because it was a really difficult project, my supervisor at Canterbury, Ren Dobson, Rachel and David at Stockholm, Peter and Sam made the nanobody in yeast, and Cesare and Maria Francesca in Italy, they do these fantastic proteolyposome measurements for us. So that's been a really wonderful collaboration. And thanks to everyone for listening. I'd be happy to take questions.
Well, first of all, James, super awesome talk. I had seen some of your work, but I hadn't seen it in such detail. I think it's super. There's a lot of stuff that I want to discuss with you later on. I'm getting excited because I want to make questions. But also, before we start the questions, I just want to say your figures, man, have always amazed me. I need to learn a lot about this from you. You really make you choose the right colors. You do the right zooms. And,
yeah, thank you very much.
And congrats on your structured paper, man. That's super, super cool.
Yeah, very proud.
Okay, I'm gonna switch to Spanish a little bit to tell the students if we have some time for questions.
Sure.
Chicos, adelante. Vamos a dejar unos minutos para preguntas. La plática del doctor James estuvo muy, muy suave. Las estructuras están súper, súper, súper suaves. Entonces, adelante, si quieren hacer la pregunta en español, pueden hacerla en español, pueden preguntar en inglés también. Si les da pena, pueden escribirla nada más y yo la puedo traducir. Adelante, Fidel, puedes hacer tu pregunta.
Hi, James. Thank you for the symposium, and what awesome figures. They are very awesome. I have two questions. First is about the figures. What program do you use to make the figures? I try sometimes to make figures, but it's very hard. So just a little bit, I want to know what program do you use?
Yeah, sure. For the protein structures, Chimera X is my go-to. And I guess just Chimera X and Illustrator, Adobe Illustrator for making the figures. Yeah, it takes a while, but I'd say the forums for Chimera X and the mailing list, if you're doing the mailing list, they're really useful to learn some of the tricks for representation. Yeah, really recommend that.
Thank you. My other question?
Wait. He also uses LeakFlood for some of his figures, and he uses this other software, which was super cool, that has like better resolution to make the
yes. The web server, I believe, called Protein Plus. link plot and protein plus for the sort of 2D. You mean the 2D ones, Francisco?
Yeah.
So this one here was from Protein Plus server. It's really easy to use.
Go ahead, Phil.
Okay, and my other question is about the CRP binding protein. When it binds with the transporter, with the membrane transporter, I watch a figure where it has some binding sites, and you made the AlphaFold structure. How does it release the transporter? Does it have a conformational change or something like that?
How did the binding protein come off the transporter?
Yeah, please.
Yeah, sure. I think, it's really hard to see, but I think that this movement up of the transporter this is I guess, what did I say here? It pushes the button of the elevator. So the operator pushes the button of the elevator, making it go up, and then the elevator going up actually allosterically opens the binding protein. So it's just a physical sort of going up and then causing that to happen. And that state where the binding protein is more open has a reduced affinity or a mismatch with the transporter itself.
So at the same time, then it push up, it opens and release the. Okay.
I think that's a nice explanation. We don't really have data for that actually, so that's our model. But we're definitely still testing that.
Thank you so much, and congratulations for your paper.
Thanks for that.
Adelante, chicos. ¿Alguien más que quiera hacer una pregunta? Tenemos algo de tiempo, entonces podemos aprovechar ahorita todas las preguntas que le quieran hacer a James. Incluso si le quieren preguntar un poquito de cómo le fue en el doctorado o en sus estancias postdoctorales. Ya recuerden que viene de Nueva Zelanda. Si le quieren preguntar ahí de Nueva Zelanda, también adelante. So we just now wait to see if there's another question from the students. I guess while we're waiting, like this is super interesting. So the P subunit has high affinity for the sialic acid, but the membrane part is super low affinity. So it makes sense. It catches it, it goes into it. How do you check the actual affinity for the membrane subunits with the P subunit?
That's a really good question. I guess the traditional thing to do would be use SPR, surface plasmon resonance, which was what we were going to do, but we started off doing spinning the proteins together in an analytical ultracentrifuge. So trying to see if we can use the centrifuge with optics inside it to watch the proteins sediment together. And those experiments were really hard because essentially no matter how much P subunit we put in, we couldn't see binding. Then binding looked to be in the millimolar regime. We also used a technique called microscale thermophoresis, MST. With that, we could get a little bit of binding, but we could never get a full binding curve because of the difficulties, I guess, of trying to treat the binding protein like a normal ligand and getting it up to high concentrations. Yeah.
Okay, so were you, like, when you were doing these experiments, were you putting high concentrations of sialic acid to make sure that the P subunit was always bound to the substrate?
Yeah, that's right. I guess we had to go up to, I think, from memory, it was around about 300 mg per mil binding protein, which is an awful lot. But I guess we didn't actually have to add that much sialic acid in because the affinity is so high. But yeah, it is really a topic of ongoing research. We're looking at things like thermophilic organisms, so organisms that live at high temperatures, and their enzymes tend to be much slower at room temperature, and the transporters as well. So we're looking at exploiting those to see if we can sort of trap some of these intermediate states.
Is it possible, because when you got the cryo-EM structure, you only got the membrane part, right? You never got the P part bound to it?
That's right.
Is it possible there's a third protein that's helping the interaction? Because I recall, for example, on GPCRs, when the substrate is binding just to the membrane part, it has like some sort of affinity to it. But if you have G protein plugged in there, the affinity is off the roof. So
yeah,
could this be like a similar situation?
That's a really, really interesting question. I think for the majority of them, no, because this is if you look at the gene structure of the operons, they're very tightly regulated, clustered genes together. The P, the Q, and the M, they're all together and they're all regulated together. So they're probably working together. There is some out there that we've sort of started to look at where there is a gene close by where there's a domain of unknown function, where there could be a helper protein. So yeah, maybe. Yeah, we'll just have to see on that one.
Okay, so I guess there's a lot of cool stuff that you can do on this, and I remember you're still working on it, so I'm pretty certain you're gonna figure it out pretty soon. Now, I want to make another question for your other carnitine receptor.
Yep.
Get to that one. So the carnitine receptor, you said if there's mutations on it, and we cannot import the carnitine, essentially you said people can die, no? So if we were to activate this carnitine receptor and we're going to be able to accelerate the binding of or metabolism of the fatty acids, you think that would be like a possibility for a new weight loss drug?
I think carnitine is taken as a nutraceutical now already. I don't know if it's got much potential as a weight loss drug, but yeah, I know some people do take carnitine like creatine as part of their diets when they're exercising to increase the rate of the fatty acid oxidation. Yeah, but I think you get a lot of it from the diet anyway. And you also make carnitine in your liver, I believe, as well. So it comes from both the diet and it's synthesized within the body. So probably not going to be as good as a GLP-1 agonist.
So what if you make a small molecule that accelerates the receptor? You know, hey, you patent that and then you retire.
Yeah, I wish.
I also, Gilberto, go ahead with your question.
Again, thanks for the talk. This is more of a general question. You mentioned that you start to train in crystallography, and then you start training cryomicroscopy. Was it challenging to change? And also, have you considered to maybe study these proteins with molecular resonance?
Yes. Great question. It was tricky to change. I guess I was really committed to crystallography, and there's a lot of theory and stuff to learn there. It's very complicated. In a way, cryo-EM can be a little bit more simple in that we don't have to do well. It's still very difficult, but the process of solving the structure in terms of not needing to get the phases is much easier. So that made it, once you started, it made it easier. And I guess in New Zealand, where I was doing my PhD, we don't have a synchrotron and we don't have any microscopes. So we have to travel to do our research, probably a lot like you guys. And Australia, where I am now, is the place where they have all these microscopes. So I guess it was definitely difficult, but it's worthwhile. What was the other part of your question? Sorry. NMR. Yes. Francisco actually had a great idea about doing saturation transfer difference NMR. So like these proteins are still too big, I believe, to really look at like structurally in the NMR, but to look at ligand binding, yeah, I think we could in the future.
Thanks.
Yeah, cool.
So if I can ask another question, if we have time. You were mentioning that you have to travel to Australia to make some experiments. How is the state of the art of science in New Zealand? Like, are PhD students in New Zealand considering to move to other places to continue their career, or what do you think about that?
Yeah,
So I want to interrupt a little bit here because I just want to let you know, James, that Alberto has lived in New Zealand.
At least for a small protein like this, where we're not really hitting the resolution where we're only just seeing these sorts of things, right? And I feel like it was, I guess the decision was that making mutants and solving those structures might be quite risky in terms of getting the resolution needed to sort of understand this. So in this way, I guess crystallography in this case would be much more valuable. The resolutions in which you can achieve the crystallography would be really useful if we could achieve that sort of 2 angstrom mark. But that's really rare in cryo-EM. You have to get quite lucky to get there. So yeah.
Okay. Well, I hope maybe in the future you try to do that, because I think like knowing how the mechanism of the disease would be super cool to know. And when you're saying it's super expensive, man, you have the Antarctica right there for you at your disposal.
Yeah, probably shouldn't complain too much. Yeah, maybe some molecular dynamic simulations would be really useful in this case as well, to sort of understand how these molecules are all moving within the protein would be really useful.
Okay. So Gilberto's looking for a place to do his PhD. Maybe he's still figuring out, but maybe now that he has seen all of this cool stuff you guys do, it might be yeah.
The weather's great. The weather's great in both countries. Maybe it's better in Australia. Maybe it's better in Australia though.
Yeah, maybe I will reach out to you in an email or
Please do.
yeah, it seems great.
Of course.
Okay. So, chicos, alguien que guste hacer una pregunta, igual les comentaba, pueden hacer la pregunta por texto y yo la puedo traducir si la quieren hacer en español. Si ya no tenemos preguntas, pues podríamos concluir el seminario. Anybody else want to make one last question before we finish up? Okay, so I guess that would be like the last question, James. So I think we'll just wrap it up. Again, Manuel is saying thank you very much for the great seminars. If you do go, James, to the next EBEX, you would meet Manuel. He's a professor at the biggest university in Mexico, UNAM. He's a career researcher. So, James, here we have, we will send you this certificate for your talk. We are so honored that you gave a talk to our university. The idea of our talks is always to like broaden the horizon for our students. Maybe as a small university, we are not especially known for our research, but I like it that our students are able to see that what great science is done in different places, and to us that we're in the other side of the world, to be able to have researchers from other places of the world and show us that they're doing like such nice science. I hope this stimulates them to think that the world is much bigger than they might think it is, and that there's so many things to learn and so many things to do. So with that said, we're gonna give thanks to Professor Alastair and Professor James for their great seminars. I just showed them the certificates that we will send them soon on the email. I will switch a little bit to Spanish to give the last part, and then I'll switch back to English. Entonces, chicos, a través de la Facultad de Ciencias Químicas e Ingeniería y de la UABC, nos despedimos de los estudiantes y del público en general, además de los profesores que nos acompañaron el día de hoy. Esperamos que les haya sido de su agrado y les sirva para ampliar su panorama a la hora de elegir su próximo paso al culminar su carrera universitaria, y para que conozcan un poco más sobre lo asombroso que es este campo de investigación de la biología estructural. With this, we will finish our symposium. Thank you so much, James. I hope we can collaborate in the future, and I'll keep in touch to see what's going on. You guys are helping me out with some experiments, so
of course. Thank you very much for the opportunity, Francisco. Really appreciate it, and thanks everyone for coming today.
All right, so everybody have a great afternoon, and hope to see you everybody again soon.
Thanks.